Hi everyone, welcome to Chapter 2 Chemistry Comes Alive. So, in this component, you're going to learn about several polymers, such as the polysaccharides, the DNA structure, the nucleic acids, the proteins, lipids, so on. So, let's go through this presentation. So, if you remember, we talked about the anabolic process and the catabolic process, and here you're going to learn about some organic molecules. So, before that, let's understand that organic molecules we categorized when they contain the carbon atom. There are exceptions, though, that is, carbon dioxide and carbon monoxides, even though it does have carbon atom, it does not considered as an organic molecule. They are inorganic molecules. So major organic compounds that is relevant with related to our study or course is that carbohydrates, lipids, proteins, and nucleic acids. And these compounds mostly are polymers. So polymers are composed of monomers, and what is a monomer is that it's a building block. So a chain of similar units or monomers combined together and formed a polymer. With related to this formation of these polymers, there could be synthesis process. That is, so if the synthesis is happening, it's called the dehydration synthesis. And when you need to break down these polymers, we call it as the hydrolysis reactions. So let's look at what we have discussed before with related to the synthesis and hydrolysis. So the first figure shown in here is that there are two monomers, two different monomers, though it's been shown here. And when they formed together, they formed a covalent bond. So monomers linked by the covalent bond. And at that point, there is a removal of hydroxyl ions from one monomer is happening. This is the hydroxyl ion, and removal of hydrogen ions from the other, and that is how the covalent bond is formed. So the water molecule is being released. And so this is a dehydration synthesis process, which is like dehydrate means lose of water. So with the formation of the molecule polymer, that it's removed the water molecule over here. With related to the hydrolysis process, the same compound shown in here, to break it down into monomers, now you required water molecule to be added into the reaction. So that is, the water molecule is going to be break down. So hydrolysis is happening. So monomers are going to be released by the addition of water molecule, because it's going to be added once the water molecule is going to break down. The hydroxyl ions of it is going to add into one monomer. The OH, the other hydrogen ion is going to add it into the oxygen, which is in free. And when you think about the reactions or synthesis and its breakdown process, that one of the examples that you would take is like formation of saccharides. Saccharides are formed by monosaccharides. You're going to learn those things later. And here is a monosaccharide glucose, the monosaccharide fructose. When they form together, the water is released and sucrose is being formed in this forward reaction. And for the sucrose to be break down, you need water incorporated into the reaction, and then it releases fructose and glucose. So this is kind of the storage process involved with the storage process, as well as when we digest sucrose as like normal simple sugars, how it hydrolyzed in our body as such. So let's look at these polymers one at a time. And these are very good multiple choice question areas that you need to understand the examples of monosaccharides, disaccharides, polysaccharides as such, and what its components, so on. So carbohydrates include sugar and starches. The chemical composition would be carbon, hydrogen, and oxygen. Hydrogen and oxygen are in 2:1 ratio, and there are three classes, that is monosaccharides. We call them as simple sugars, and this is the smallest unit of the carbohydrate. Then you encounter the category disaccharides. These are two sugars, and the polysaccharides, these are many sugars. So the polymers, they're made up of monomers of the monosaccharides. So let's look at them in depth. So monosaccharides, they are simple sugars. They're containing 3 to 7 carbon atoms. So the common formula, the general formula is, within the parentheses, it is this carbon, hydrogen 2 molecules, oxygen, and there is an N number. And the N number is the number of carbon atoms. For instance, if you take glucose, you have the N number of 6 carbon atoms. So you multiplied it carbon with 6, so you get 6 carbons, 12 hydrogens, 6 oxygens with related to the glucose structure. The monomers, the monosaccharides are the monomers of the carbohydrates. There are important monosaccharides, pentose sugars. The categories are the ribose and the deoxyribose examples. Also the hexose sugars. These are the glucose and blood sugar. The lay language you used as the blood sugar is referred to the monosaccharide hexose sugar, which is, we all know, that is the glucose. So underneath you would see the structure of glucose, fructose, the galactose, and also the type of pentose, the deoxyribose and ribose. And how you say that it's pentose, you would see the shape of the structure. You have this 1, 2, 3, 4, 5 sides. Here is the hexose, and then you would see the 6 carbon atoms over here. You would find 5 carbon atoms over here. Let's look at disaccharides now. Disaccharides refer to as double sugars. So two monosaccharides combined together and formed disaccharides. They are too large to pass through the cell membrane. And the important disaccharides that you encounter are the sucrose, maltose, lactose. They are formed by the dehydration synthesis, and examples is being given here. When you combined the glucose monosaccharide, fructose monosaccharide, it forms the sucrose disaccharide and release the water molecule. And here it's been shown down here, the other disaccharides, the maltose and the lactose formation. Then polysaccharides. Polysaccharides, of course, it's the polymers of the monosaccharides. This is also formed by the dehydration synthesis of many monomers. And the common ones that you encounter would be the starch and the glycogen. Starch is a polysaccharide that you commonly found in plants. It is the storage form of carbohydrates for plants, and glycogen is found in animals, and that is the storage form of carbohydrate in animals. And these are not very soluble because of its big, large structure. So here in this figure, it shows the glycogen molecule, which you would see that the glycosidic bond. Of course, all monosaccharides, they formed glycosidic bonds when they form either disaccharide or polysaccharide. So these are the glycosidic bonds that you encounter in these polysaccharides, and you would see some polysaccharides are branched, as you can see in here, and sometimes some polysaccharides are not branched. So here branching means you have some side chains which is attached to the major structure. So here is a branched polysaccharide, which is a glycogen depicted in this figure. Now let's look at the lipids. So the chemical composition would be that it also contains carbon, hydrogen, and oxygen, but less than in carbohydrates, and sometimes it can contain phosphate or phosphorus. Phosphorus. So this is insoluble in water, lipids, and there are many types: triglycerides or neutral fats, phospholipids, steroids, and icosanoids. They all come under the category of lipids. So let's look at triglycerides or neutral fats first. So we use the word fat when it's solid in physical status, or when it's in liquid, we call it as oils for neutral fats. And this is composed of 3 fatty acids, and they are bonded to a glycerol molecule. So as you can see in this figure, this is the glycerol structure over here, and the 3 phospholipid, the 3 fatty acids here. You would see the first fatty acid chain here, the second fatty acid, and the third fatty acid. So this is a synthesis process. So as you can see, it would produce 3 water molecules in the formation of a triglyceride or a neutral fat. So you would see the glycerol structure over here, the 3 fatty acid bonded to the glycerol structure over here. And the major functions of neutral fats would be storage of energy and provide insulation, and also the protective function and the cushioning function in some places, especially over the buttocks area where you can find lots of fats, so on. Triglycerides, it can be constructed of sometimes saturated fatty acids or unsaturated fatty acids. In saturated fatty acids, all carbons, they're linked through a single covalent bond, and this would result in maximum number of hydrogen atoms, so which refers to as saturation with hydrogen atoms. So these are mostly solid at room temperature. The examples that you would encounter is animal fats, butter, and other saturated fatty acids. With related to the unsaturated fatty acids, the difference is that the carbon atoms, they are linked with double bonds, and this would result in reduced number of hydrogen atoms, and that is unsaturated, and mostly they are in the liquid form, mostly plant oils, olive oils, they comes under unsaturated fatty acids. And trans fat, these are modified oils. So as you all know that this is an unhealthy component. And omega-3 fatty acids, of course, it would help for the heart, and that is a useful component for healthy heart. With related to the modified triglycerides, the common example would be the phospholipids. So phospholipids, it is composed of a glycerol and 2 fatty acids instead of 3, but it does have a phosphorus containing group, so that's why it's the word phospho come into that. And with phospholipids, there are two regions, the head region and the tail region, and they do have different properties. The head region is a polar region and it is attracted to water. And this is mostly where the phosphate group region. And the tails, they're non-polar. As you can see over here, there are two fatty acid chains and they are repelled by water. So that is hydrophobic. And the head region is called the hydrophilic region and the tail region is called the hydrophobic region. And if you already know, or as you move on with the course, you will understand that phospholipids are very important component of the cell membrane structure, and this hydrophilic and hydrophobic component is helpful for the arrangement of the plasma membrane phospholipids. The other category under lipids would be the steroids, and steroids consist of 4 interlocking ring structures, as you can see in the figure below. The common steroids would be cholesterol, vitamin D, and some steroid hormones, estrogen, progesterone, testosterone, and also bile salts. Most important steroid is the cholesterol. It is a building block for vitamin D and for steroid synthesis and also for bile salt synthesis. And cholesterol is also an important part of the cell plasma membrane structure. The last group is the eicosanoids, which is derived from a fatty acid, arachidonic acid, and it is found in the cell membrane. The most important eicosanoid that you encounter would be the prostaglandins, which play a role in the blood clotting process, and also they're involved with control of blood pressure, inflammation, as well as mainly for labor contractions. Now let's look at the other polymer, which is the proteins. Proteins, they comprise of 30% of the cell mass, and it does have lots of functions. So sometimes it forms structures, sometimes it works as enzymes, sometimes it do the function of contractions, so on. The chemical composition, other than the carbon, hydrogen, oxygen, it does have nitrogen and sometimes it does have sulfur and phosphorus. The monomer which makes proteins would be the amino acids, and 2 amino acids held by a specialized bond called the peptide bond. So in one structure of the protein, the primary structure of the protein, you would encounter lots of peptide bonds because lots of amino acids combine together and form the primary structure, and to form that you have to form peptide bonds. And depending on the shape and the function of the proteins, there are 4 structural levels, and let's look at them. Before that, let's understand the amino acids and the peptide bonds. So, as I mentioned before, proteins, the monomer would be amino acids, and so far, there are 20 types of amino acids involved with formation of proteins encountered in the human body. These peptide bonds are type of covalent bonds, and they contain the amino acid. If you take individually, it contains the amine group as shown in this figure, the amine group, the NH2, and the acid group, the COOH group, and it therefore the single amino acid or proteins, they can act as either acids or base. And the only difference, because if you see, this is a common structure that you would encounter in each amino acid. The difference is provided by the R group. So therefore, in 20 different amino acids, the R group would be differ. The rest of the structure would be similar. So here it shows the formation of the peptide bond, which is due to the dehydration synthesis. Two amino acids combine together and it releases the water molecule and form the peptide bond as shown in here. And to break this peptide bond, as we mentioned before, the hydrolysis needs to be happen. So water molecule is incorporated and release the relevant amino acids. So as I mentioned before, let's look at the 4 structural levels of the protein. And the first one would be the primary structure, which is the linear sequence of amino acids in order. The secondary structure would be the how primary amino acids interact with each other and they formed hydrogen bonds. And based on their organization, there are alpha helix coils, which resembles like a spring structure, or it might arrange as some pleats or pleated sheets, which is resemble like an accordion ribbons. The third structure would be the tertiary structure, how the secondary structures interact together, and the quaternary would be how 2 or more different polypeptides interact with each other. So here you would see the primary structure, the polypeptide, because you see they formed the peptide bond. So there are lots of peptide bonds. So that's why it's called a polypeptide. As I mentioned before, the primary structure is a linear sequence of these amino acids. And you formed a covalent bond in the as a peptide bond. And with the secondary structure, you would see obviously the alpha helices like a spiral and the pleated sheets like an accordion ribbon. You would see a new bond, which is the hydrogen bonds, which happens between those atoms. And actually with the denaturing process of the proteins, the hydrogen bonds are the ones which are going to break down. Thinking about the bond strengths, thinking of ionic bonds and covalent bonds, the covalent bonds are stable than the ionic bonds. That's I thought to mention. Maybe not relevant with the hydrogen bonds over here, but just for the sake of your knowledge. So here, as you can see, continuing, you see lots of hydrogen bonds formation, and that's why the primary structure is now formed into the secondary structure. And with relate to the tertiary structure, the secondary structures, the alpha helices or beta pleated sheets, they are folded up more and then form some kind of a compact globular molecule. And these structures, the molecule is now held together by intramolecular bonds. So inside there are much more intramolecular bonds is going to be formed, and that would help to form a globular structure, and we call it as a tertiary structure. With related to the quaternary structure, 2 or more polypeptides. If you think this is still a single polypeptide with related to this tertiary structure, in quaternary structure, you would now encounter 2 or more polypeptides. So in addition to intramolecular bonds, you get intermolecular bonds. So that's how the quaternary structure would be formed. So examples for quaternary structure would be the hemoglobin molecule. So based on the shapes of the proteins, we can categorize the proteins into fibrous or globular. Fibrous proteins are structural proteins. They are strand-like, they're water insoluble, and they're much more stable. Most have tertiary or quaternary structures, and because they're much more strand-like and stable, they provide some mechanical support and tensile strength. Examples that you would encounter would be the keratin, elastin, collagen, and certain contractile fibers. They comes under fibrous structural proteins. The globular ones are much more functional proteins, and they're much more compacted, spherical, and they're water soluble, and they're sensitive to environmental changes. So they do have the tertiary and the quaternary structure, and based on their structure, they have some functional regions. We use the word active sites for them. Examples would be the common one, enzymes, and also hormones and antibodies. They do have these active sites, or some kind of a site where the ligand can come and bind and react with those molecules. With related to the protein, we need to talk about the denaturing process. With the denaturing process, the globular proteins is going to unfold and lose their functional 3D shape. The hydrogen bonds are going to be broken down. The fibrous proteins, however, are much more stable to the denaturing process. With related to the globular proteins, the active sites is going to now become deactivated due to the denaturing. And the denaturing can happen due to decreased pH or increased amount of temperatures, and usually the reversible if the normal conditions is going to be restored, but if it goes to extreme conditions, it cannot reverse. For example, the irreversible process would be like if you take an egg is made from albumin, and when you heat up, you would see the nature of the egg white is going to be changed, and then it's going to be hardened. But if you're going to be try to reverse it back, it would not happen, because now the structure the bonds have been broken down. But certain enzymes, like for certain times like hyperthermia, or as such, like the enzymes would not be functional, but if you quickly drop your temperature down and maintain into normal temperatures, then the enzymes there's going to be function as usual. So that is the reversible process. But irreversible, the egg white example, cooking an egg would be a nice example. So with related to these globulous proteins, we have to discuss some amount with related to the enzymes and its activity. So enzymes, they are globular proteins, and it is the biological catalyst in our body. What is a catalyst? Catalyst is a compound which regulate and increase the speed of the chemical reaction without getting used up in the process. So it just enhance or increase the rate of the reaction, but it does not consumed into the reaction process. It does not become part of it. And the major important component of catalyst, or why we need catalyst, is that it lowers the energy needed to initiate a chemical reaction. So you might have heard in chemistry, for each reaction, you have to have an initial energy. With related to the enzymes, this energy is going to be lowered down. Without the catalyst, you need to have lots of energy, initiation energy, to start the reaction and to continue. But with related to the presence of catalyst, you just need to have less amount of energy. With the presence of catalyst, it leads to increase in the speed of the reaction, and also it allows millions of reactions per minute to happen. So that's why we can be active as usual. Otherwise, we would be much more lethargic. So characteristics of enzymes, most functional enzymes, they are referred to as the holoenzymes. And holoenzymes, it consists of two parts: the apoenzyme, which is the protein portion of the enzyme, and the cofactor, that is the metal ion or coenzyme, the organic molecule, often this is a vitamin. So the apoenzyme part and the cofactor or the coenzyme. Either it could be a cofactor or coenzyme goes with the apoenzyme component. Enzymes, they are specific and they act on very specific substrate. For instance, the enzymes works for carbohydrates cannot work for lipids. So it's specific for carbohydrates. How we recognize enzymes, there's a little trick that when you see the ACE word end in a word, then you often know this is an enzyme. Examples: hydrolysis, oxidases, peptidases, dehydrogenases, polymerases, all are enzymes. So let's see the enzyme action now. The chemical reactions needed for activation energy is going to be lowered down by the enzymes. Here you have, it's been shown like two pictures which shows the chemical energy needed without the enzyme to initiate a reaction. And if you compared the first picture to the second picture, you would see that the energy required for the second process with the enzyme is much more lower. So with related to the enzyme action, there are 3 steps involved with the enzyme action. The first one would be the substrates are going to bind to the enzyme active sites, and this is a temporal process, and we called formation of the enzyme substrate complex. So here the orange color oval shaped compounds are the substrate, and enzyme is been shown as in purple. You see the active sites, 2 active sites, and when the substrates are bound to the active sites, we call the enzyme substrate complex. And then once this formed, complex is going changes, rearrangement is happening, and it may be formation of bonds or breakage of bonds like that. So here you would see a formation of a bond over here with a rearrangement process. And then at the last step would be that the enzymes product is being the product is going to be released from the enzyme, and it's open up its active sites once again. So lastly, we need to talk about the last molecule, which is last polymer actually, which is the nucleic acids. So nucleic acids are very important because they are the building blocks for our genes, the DNA. Nucleic acid, the chemical composition would be carbon, hydrogen, oxygen, nitrogen, and phosphorus. And these nucleic acids are the largest molecules in the body. Nucleic acids are polymers, and the monomer which form these polymers are the nucleotides. So if I ask like what are the components of nucleotide would be, you need to know that it's the nitrogen base, a pentose sugar, and a phosphate group. So these 3 components, nitrogen base, a pentose sugar, and a phosphate group, formed nucleotides. There are 2 major classes: deoxyribonucleic acid, we call it as DNA, the common word. The ribonucleic acid, we use the word RNA in short form. DNA, it holds the genetic blueprint for the synthesis of all proteins. It is a double helix, and it is located in the cell nucleus. And also, if you take some organelles in mitochondria also, there are part of DNA is presenting, but for the genetic purposes, the genes or the blueprint for the individuals is in the nucleus. Nucleotides, it contains a deoxyribose sugar. I highlighted the deoxy because it's not just ribose sugar, it's the deoxyribose sugar and phosphate group and one of the 4 nitrogen bases. So you need to know the nitrogen bases, the 4 nitrogen bases. The purines are adenines and guanine. The pyrimidines are cytosine and thymine. So the 2 major categories are purines and pyrimidines. Under purines, you get the adenine nitrogen base, guanine nitrogen base, and under pyrimidines, you get cytosine nitrogen base and thymine nitrogen base. So the bonding of these nitrogen bases happens from strand to the opposite strand in a very specific manner. In the DNA structure, we call it as the complementary base pairing rule. So A is always paired with T with 2 hydrogen bonds. G, guanine, is always paired with cytosine with 3 hydrogen bonds. So here, this structure is very important. Sometimes you might see these in your exams as well to identify the structures and to label them because in your exams, we need to test your anatomical knowledge. So sometimes we use figures for your exams. So here you would see the structure in a little bit more magnification. These are the hydrogen bonds that I mentioned before. So here you would see this is the ribose sugar and the phosphate group alternatively, and these are complementary. And this is the double helix of the DNA structure. The color coding would help you to differentiate the different nitrogen bases. You would see here adenine is forming 2 hydrogen bonds. See 2 lines. The lines are formed from the dotted ones. With thymine, with cytosine, you see 3 hydrogen bonds, that is 3 dotted lines with the guanine, like that. So it's easy when you're given to identify these nitrogen bases. Look for these hydrogen bonds and see like how many hydrogen bonds, and you just need to know the rule. Other than the DNA, the other important nucleic acid would be the RNA. And the difference would be that RNA is a single-stranded and it's mostly active outside the nucleus. DNA is in the nucleus. RNA is mostly outside the nucleus. RNA contains ribose sugar, not deoxyribose. Thymine nitrogen base in DNA is now replaced with uracil. So RNA, it does not have thymine, it does have uracil. And there are 3 varieties of RNA which carry out the DNA for the process of protein synthesis, that is the messenger RNA, transfer RNA, and the ribosomal RNA. So all 3 RNA are involved with the protein synthesis process. Other than DNA and RNA, there's another type of nucleotide, which is the ATP, where glucose is broken down, and when the chemical energy is released, when glucose is broken down and when the chemical energy released, that this energy released is going to be caught by a molecule, which we called it as the ATP molecule. And therefore, once it's caught, it's going to store that energy and therefore we can function, because when we function or move, digest, for all energy requirement process, we use that stored energy in that ATP molecule. So ATP stands for adenosine triphosphate. ATP is directly powers the chemical reactions in the cell. It is offers the immediate usable energy needed by the body cells. So ATP, when it's released its energy, it forms the ADP, adenosine diphosphate, and a phosphate group in addition to its energy. To form ATP, you remove water molecule from this reaction, the reverse reaction. To form or to release energy, you have to hydrolysis. It's a hydrolysis reaction where you release the energy from the ATP. So let's look at the structure of the ATP. Adenine containing RNA nucleotide. So it's a RNA nucleotide with two additional phosphate groups. This is the last phosphate group when you release the last phosphate group, you formed the adenosine diphosphate, you would see. So these two these two bonds, the high energy phosphate bonds, are the ones which store energy. So you can break down this phosphate bond, and release phosphate group, and it will end up with adenosine diphosphate and a phosphate group and energy. You can further break down this molecule, the ADP, then you would result in AMP, adenosine monophosphate. Mono means one, so you would end up only with this structure because all those two phosphate bonds are being broken down. So this is the end of chapter 2. I will meet you in our next lecture. Thank you.